It's my great pleasure to introduce to you my postdoctoral work in the past few months, which is using human retinal organoids to understand retinal ganglion cell axon outgrowth. We use eyes to get most of the information from our environment, so definitely vision is one of the most important senses for our life. As we can see, the light goes through the cornea, lens, vitreous, then retina, where it converts light signals to electrical signals. The electrical signals go from photoreceptors to interneurons, then retinal ganglion cells, RGCs for abbreviation. The RGC axons extend and bundle together to form optic nerve like a bridge to convey visual information from the retina to the brain. These RGCs are vulnerable. The death of RGCs causes the degeneration of the optic nerve. The representative disease characterized by that is glaucoma, a leading cause of vision loss in the aging population. To preserve or restore vision through clinical therapies such as transplantation, it is critical to prevent RGC death and to stabilize RGC axons. A breakthrough technology is the development of using human retinal organoids to study human biology. The Johnston lab pioneered the use of retinal organoids to determine mechanisms of human retina development. The retinal organoids are differentiated from human pluripotent stem cells. They are similar to developing human retinal tissue in terms of architecture, types of cells, and developmental stages. It's an in vitro system, so we can use them to perform genetics. We can also induce RGC axon outgrowth from cut organoids on matrigel-coated surfaces. Here comes back to my main point. We need to prevent RGC death and to stabilize RGC axons. So what conditions promote axon outgrowth and RGC survival? Is it possible to use human organoids to set up that condition? If that could be achieved, we will be able to ask further questions, such as how do RGCs and other neuronal subtypes interact in those conditions? What molecular factors facilitate axon outgrowth, RGC survival, and cell-cell interaction? Next, I'm going to introduce how to establish the organoid interaction assay for that condition. Organoids are cut, placed in proximity, and cultured to monitor axon outgrowth and interaction. We can see that the axon outgrowth is induced. The two cut organoids form an interaction area where RGCs and other neurons intercalate with one another. Then, how to visualize that the axons are promoted, that the RGCs are stabilized, and that the interactions among cells are increased? I detached one organoid by an applied force, such as slight vibration. As we can see, the axons remain connected. The detached organoid is pulled closer to the affixed organoid. Then, after a couple days, the detached organoid formed new axons, and the organoid is stabilized again. Now that I have established the organoid interaction assay, next in the future, I will examine how RGCs interact between organoids. The Johnston lab has successfully grown organoids that fluorescently mark RGCs and their axons. I will conduct the organoid interaction assay with one fluorescently labeled organoid and one unlabeled organoid. I will visualize and track the morphologies of the axons from labeled organoid. Then, in order to investigate genes responsible for stabilizing the axons in the interaction area, I will conduct RNA-seq to generate a list of candidate genes that contribute to interaction area formation, perform in situ hybridization or immunohistochemistry to assess gene expression patterns, and use CRISPR to knock out candidate genes and assess whether they regulate axon stability. To sum up, our work advanced human retinal organoids as a model for revealing mechanisms of RGC survival and axon stabilization, with implications for glaucoma disease treatment involving organoid-based transplantation. With that, I'll thank Johnston Lab at Johns Hopkins University, especially Bob, for giving me such a wonderful chance to figure out a way to test organoids interaction and axon stability. I also thank all lab members for helping me initiate my project. We also thank Zach Lab at Johns Hopkins University for generously providing the Brain3B TD Tomato human stem cell line. And finally, thank you all for making this year's meeting online successful and for your attendance.